So, the cells actually, obviously you start, let's say, with a zygote, which is a fertilized egg, right, from one sperm, one egg. And that's got it's got 2 copies of each chromosome, so it's 2N. The gametes themselves are each 1N, so the one sperm, there's 1 copy of each of the 23 chromosomes, and egg, same thing. Okay. Now, when we're developing, or when any organism is developing, the cells obviously start splitting and multiplying many, many different times. And that's all controlled by some very specific genes that control development. And then, once you've got an individual who's mature, some cells keep replicating themselves as needed. So epithelial cells, skin cells, there's usually some being replicated every day. Red blood cells, I don't know, a couple months they last. And then muscle, much longer. Neurons, almost never. We lay down new neurons but for memory, but once the neuron is there, it's pretty much what we've got. So, one of the things we're going to learn about is cell division and replication. And then we'll learn about what goes crazy when people have cancer. So this is actually a cell that's in the process of replicating into 2 different cells. That's during what's called anaphase, and we'll get into the specifics of that. But cells do this all the time. So when cells replicate themselves, in terms of the genetic material, obviously the new cell is going to need its full complement of all the chromosomes, right? Because if you remember correctly, all cells have the complete genome inside those cells in the nucleus. Unless we're talking about bacteria, and then bacteria and archaea don't have a nucleus or any organelles. So we have these 2 chromosomes here, A and B, or A and, let's say A and A, yeah, right. One's from dad. This one, and then one's from mom. This one, right? And then before a cell is going to divide, you need actually to make 2 copies. So you replicate the DNA. Okay. Now, the next thing that you need to do, of course, is you need to condense the DNA, and we'll show you why. So the DNA in the nucleus is usually in a state of being uncondensed. And so why might most of the DNA be uncondensed? And how might that relate to gene expression? So we just learned about gene expression. So do you think, let me show you. So why do you think it's like that in terms of gene expression? What?
Easier to produce protein?
Mm-hmm. So it's easier to be read. Normally what happens is in condensing the DNA, it's very tightly packed. And the different loci, gene loci, are not really open to being turned on or off or expressed or anything like that. So for the most part, like this DNA is kind of actually partly condensed, but not much, so that the genes can be expressed. Now, in terms of replication, though, it's not very easy to divvy up the DNA this way when it's looking like this. So it has to be organized in a way, and that's what happens with condensing the DNA. Okay. So what happens is we condense the DNA so it's easier for replication. And this is what it looks like. Each of these are chromosomes. Okay, these are homologous chromosomes. Each of these individual ones is a chromatid. And one of them is from mom, and one of them is from pop. As you can notice, all of these different chromosomes here, some are longer, some are shorter. They all have what's called a long arm and a short arm, as you can see. So right here is a long arm. Here's the short arm. And then right in the middle here, actually shows it right here, is a centromere. So here's a cystic chromatid, by the way, and here's the other cystic chromatid. And then the full chromosome is like that. So DNA has to get condensed before you can actually replicate the cell. And then, of course, you have to divide the DNA into two. So you've already replicated the DNA, so there's twice as much as there normally would be. You've now condensed it. And then now you're going to divide the copied DNA, the two copies, into two, and it will produce two identical daughter cells. And that happens during cytokinesis. So daughter cells are produced, they're identical. Or they should be. There might be a mutation there or something like that, but in general, you create an identical daughter cell. Okay. Now, we're referring to this is the cell cycle. And cells go through this cycle. Actually, most of the time, cells are actually in the G1 phase. So they're mature cells. They're doing what they're supposed to do, whether they're neurons or muscles or whatever. Now, if a cell is actually going to replicate, it's going to get a signal at some point, and then they're going to enter what's called S phase, as synthesis. And that's where the DNA is copied. So now you have two full copies of DNA. It happens like, if you remember before, when we looked at this, the DNA unzips, and then a complementary base pair is added to it, so you have 2 full copies of each DNA chromosome. So all the material is copied then. And then, of course, before the whole thing starts in terms of creating 2 new cells, you need enough energy and resources for that to happen, and so you have this cell growth and preparation for division. Now, if you didn't do this, you'd probably end up with a cell that really, there wasn't enough energy, it probably wouldn't work very well. And during this period of time, also, there's other components of the cell. If you remember about the organelles, besides the nucleus, there's mitochondria, a number of other things in a eukaryotic cell. And those have to be copied as well, because each cell is going to need a full complement of mitochondria and all the other cellular machinery for turning the RNA transcript into a protein. So we'll take a look at this. Okay, so as I said, most cells generally stay in G1 unless they need to be replicated. If they need to be replicated, there's a signal. So we talked about different functions for proteins. Some of those proteins are signaling proteins that initiate this process. And there's also receptor proteins on the cell that receive those signals and then tell the cell, okay, let's start the machinery up for replication. Now, in terms of mitosis, I'll go to that movie in a second. Well, actually, I might just do it now. Those different phases, so you first start, you're in G2, and then in early prophase, the DNA condenses, the nuclear envelope disintegrates, and then you go into, start moving these things along, these duplicated chromosomes, and they separate into 2 different areas of the cell, and then that's the center of where each new nucleus is. So look at this. Okay. So this is a plant cell. Let's see, where am I? Here we go. Okay. So during prophase, of course, you can actually see the DNA condensing. This is a complex called chromatin. And then later in prophase, they actually condense. The nuclear envelope is actually falling apart. The duplicated DNA, this is the metaphase plate. And then this is anaphase. Now the longest phase is actually, I think it's prophase. And then finally, this is telophase right here. And the cleavage starts and you've got 2 different daughter cells. Okay. And so here's what cytokinesis looks like. Plants actually have a cell wall, whereas animal cells don't. They have cell membranes, but not a cell wall. So you can see how an animal cell starts to cleave, and you end up with 2 daughter cells either way. Okay. So, functions of a cell, obviously reproduction, in other words, reproduction of the whole organism. Then there's growth and development. And then there's also tissue renewal for anything that might happen that needs to be replaced. So why might cells actually divide? Can you think of an instance where it's necessary for a cell to divide? What happens when you,
Recovering?
huh?
Recovering?
Recovery?
Yeah.
Okay, good. So, okay, good. Excellent. Any other reason? What about how long do you think cells last? They vary, right, in terms of their lifespan. So, like anything else, cells wear out. Okay. So that would be taken care of during the growth and development phase. The same thing with the recovery from and tissue renewal. Let's see. There's a number of different other reasons. They'll come to me. So, yeah. So cells have different lifespans. Epithelial cells, skin cells, like I said, days, months. Organs and muscle cells, a lot of them will last for years. Neurons, almost never. And as you age, of course, being able to, replication kind of, you start losing the ability, cells lose the ability to replicate over time, and that's why one of the reasons we age. And then there's another couple of reasons why cells divide. They're lonely. But also, cells can go rogue. If a cell goes rogue, what does that sound like?
A virus?
No, but that virus is taking over a cell, okay. Any other process that might...
Betray the body.
Huh?
Betray the body. Betraying.
Betraying the body. Okay, in what way?
Like it mutates differently.
Okay, and when you have a cell like that, what kind of cell is it?
Cancer.
Yeah, exactly. Okay. Now, of course, early in development, cells are differentiating. So you start out with a single zygote, they differentiate, they replicate, differentiate. There's some cells that produce limb buds. Some parts of the tissue develop into organs, others into blood vessels, all that kind of stuff. Okay. Now, the thing is, we just mentioned cancer. Occasionally, something goes a little haywire, and we end up with a cell that's cancerous. We'll learn exactly what that means. Onco refers to cancerous. So I really don't like the name proto-oncogene. What this says is, oh, these are genes that turn the cell into cancer. Well, they're cells that need to replicate at various times. Sometimes there will be, let's say, a mutation in a cell, and that cell that's a normal cell will eventually, or a number of mutations, will eventually become, those oncogenes will actually be on or off when they shouldn't be, and you'll end up with a cell that has uncontrollable replication, which is what cancer is. So normally there's this whole signal about when to divide. There's also what are called checkpoints all along the way to make sure that the cell, when it's replicating, is following all the instructions. Okay. Now, for us to, our whole multicellular structure to work, all the cells have to be doing their jobs and staying in their place and not acting like a bunch of pirates. The thing is, if you think about it, cells can, of course, start acting like pirates. But usually what happens is there's these processes that stop them from doing it. Okay, they're actually called tumor suppressor proteins that we'll get to more specifically. But this is the kind of thing that could happen in any multicellular organism. In other words, cancer is only really found in, obviously, in multicellular organisms, whether it's a plant, an algae, an animal cell, whatever. In fact, the things that cause cells to kind of go off on their own could actually happen in a single cell. So let's say a bacteria still has to go through replicating its DNA, having enough energy and resources to do it all. And you could see, let's say there's a mutation that makes the bacteria start replicating when it's not really ready to. Okay, so in general though, we think about cancer in terms of it's a cell that's kind of gone out of control. Okay. Oh, come on. Okay, good. Okay. Now, when we were studying evolution, so selection acts on what? The population or the individual? Remember? Doesn't matter if you're wrong. Speak up. No. It acts on the individual. So those individuals that have characteristics that help it survive and reproduce, those are the ones that then become more frequent in the population. Therefore, the population then evolves. So selection always acts on the individual. Some individuals are born with certain characteristics that help those individuals survive and reproduce. And then the population changes in response to the fact that those individuals that have those characteristics become more common, while the ones that don't fade away and go extinct. Okay, so what does this have to do with cells? Well, you can actually treat a cell the same way as you can an individual. Okay? And you can have a population of cells as evolution of those cells. Okay? So, let's see, let's do this. Okay, here's our cell. And there's another cell, and another cell, and another cell. And let's say okay, but this one actually gets the green light. Okay, it's got a trait that allows that cell to reproduce continuously. Okay? When we first start, we have this whole little population of cells, right? And it's made up of all these different kinds of individuals, but eventually you'll end up with mostly these cells that are now dividing. So these cells, in terms of the population of cells, actually has higher fitness. Now you might say, higher fitness is going to kill the individual that has this cancer, right? How can that increase its fitness? Well, it has nothing to do. In terms of it's the fitness of the cell, in terms of the other cells, it's reproducing more, so it's more successful. It has higher fitness. The fact that it's going to kill the individual that it's inhabiting and itself eventually has nothing to do with, in terms of what cell is going to make most of the future population. Okay? So you can actually think of selection happening at other levels besides just the individual organism. So this is kind of what happens. You have a cell, and it replicates itself so that eventually it creates a big population that's identical to itself. That is now a tumor. And furthermore, not only that, but there'll be anyway. Here. Okay. You have other cells now that also are mutating. There's more mutations. And so this tumor, even though it's made predominantly of the blue, right? There's also some greens in here and all that. Okay, so I want you to start thinking about these are individual cells, the ones with a mutation that's beneficial to that individual cell and not forget about the rest of the other cells in the body. That's the one that's going to leave more offspring. And that's why tumors actually evolve. So cancerous tumors, it's not like, oh, you get a cancerous tumor. Sometimes you do. You have a cancerous tumor that there's very little mutation in it. It doesn't really go anywhere. You can deal with it. Other times, though, as the cells replicate, they often acquire other mutations, some of which might make that variant even more likely to divide and conquer. Now, as that woman explained, we have a system to keeps most cells from going rogue. In other words, becoming cancerous. And it then suppresses those that don't act properly. Okay. So, as she mentioned, usually it requires a number of mutations to actually happen to have a cancerous cell arise. Some of these mutations can be inherited, but most of these cells do not happen in germline cells. If you remember, germline cells are the ones that give rise to gametes, whether it's a sperm or an egg, okay? Cancer usually happens in somatic cells. See, our muscle cells, or our kidney cells, or our brain cells. These are somatic cells. They don't give rise to a new individual. Only the germline cells do that. So most of this might happen because when a cell replicates, sometimes it can get the wrong signal and start dividing uncontrollably, but in the most part, there's a suppressor system that stops that from happening. So the difference I want you to understand between somatic cells and germline cells. So during your lifetime, you can have cells that develop mutations when they replicate. A lot of those times, those replications, nothing really changes very much. But sometimes they do. And then you have uncontrolled cell replication and growth. So these are happening during the somatic cells. Now, there are certainly some mutations that end up in the germline cells that can give, once those germline cells have created an individual, those inherited variants can actually give tumor formation a head start. Okay. So, is it genetic? Well, actually, it's always genetic. Cancer is basically a genetic disease. But is it genetic in terms of being heritable? Only sometimes. It has to, for that to happen, then you need certain variants that lead to an increased probability of a cell going rogue and being cancerous. Is it environmental? Yes, because remember, when we talked about this, traits are not just genetic, which proteins are made. They're in response to certain environments, right? So, if we remember, like lactase, that mutation. Now, normally, that mutation for producing lactase into adulthood would be selected against because you're producing an enzyme that's not needed anymore. But in terms of the new environment, if you've got an environment where you have access to milk as an adult, and being able to digest milk gives you certainly an advantage in terms of being adaptive, right? Okay, so it's always gene by environment interaction that's going on in terms of what the phenotype is. Is it contagious? Okay, generally no. What does that mean? Okay, well, there are no cancers that are contagious for humans, but it turns out dogs get kind of a mild sort of cancer that they can infect each other with. And their saliva and things like that, cancer of the mouth. Rarely has any effect in terms of survival or anything like that. However, there's a marsupial in Tasmania, which is a small island off the coast of Australia, and there's something called the Tasmanian devil, and they're mid-sized carnivores. And recently, probably within the last 10 years, they developed a cancer of the face and the neck that is contagious. And you know, when you go to a movie and there's nice love songs and people are supposedly making love, you know, there's none of this grunting and shit that normally goes on. It's pretty much, oh, it's all beautiful. Well, Tasmanian devils, they're into hard sex, okay? Both sides. So, if you've ever seen, you can go online and watch Tasmanian devils mating. It's pretty amusing. In fact, it's gruesome. So they fight and they tear and all this kind of stuff. Well, they actually have a cancer too. Unfortunately, it's a lethal cancer. And once they get infected, these cancers, so they transfer cancer cells from one Tasmanian devil to another, and eventually it kills those individuals. They've now got a recovery plan where they found individuals, of course, who aren't infected. They move them to another smaller island offshore. And then there's also, it turns out, which you might actually expect, like any infectious disease, some individuals are more resistant to being infected by a bacteria, archaea, or this cancer cell than others. And it looks like there's a mutation where some individuals are not susceptible to actually getting this cancerous cell. Okay, so that'll probably spread through the population, and it'll be okay. It also turns out that these contagious cancers are not uncommon in a lot of bivalves, like clams. That was just recently discovered. Okay. Is cancer on the rise? Well, why might there be more cancer now than before?
Environmental factors
Mm-hmm.
and lifestyle choices.
Mm-hmm. What kind of environmental factors?
Pollution,
Huh?
pollution, radiation.
Right, okay. So, there's a lot of chemicals that human beings have created that are mutagens and actually and carcinogens. And so, it's likely, and it's been shown, that there have been some cancers that were caused by that. Okay. Now, remember, cancer is basically part of the package for being a multicellular organism. So, there's always been cancer. There are carcinogens from things that you eat, carcinogens from things that you smoke. Ultraviolet radiation is a carcinogen, okay? And so there are certain cancers that have been fairly constant over time, and there's others that are rather new that perhaps we didn't see before. So that's why I say perhaps. Oops. Okay. So, yeah, I mean, we have an immune system that actually is designed, if it wasn't there, all multicellular organisms would be overrunning cancer in no time. Now, there are different lineages that have different immune system, cancer suppression systems, like those between animals and plants, are very different. Of course, let's say you have cancer, it's a treatment worse than disease. Well, we'll say okay. Let's put it this way. You would never want to go get cancer therapy just for the whim of it, on a whim, right? Now, there are some cancer treatments that are pretty benign, but a lot of them are not. Okay. So this is how it happens, of course. When a gene is replicating, remember, forget about inheritance in the germline cells. When a gene, it's the same sort of thing, but when a cell is replicating, and you might end up with a mutation. So you might end up with a complementary base pair that's the wrong base pair. An A might turn into a T. Okay. Now, remember, it's not, in terms of gene expression, it's not generally on or off. It's a matter of how much. And so sometimes you'll have a gene that should be producing a lot of protein, and it's not. Perhaps that's the tumor suppressor proteins. Or vice versa, you have a gene that should at that point not be producing any protein, and it is, and so then you have uncontrolled replication. And so what will happen is with these mutations, it'll alter the function of the encoded protein, right? Now, in most cases, it'll probably make it so that that protein is not working. In fact, or rather, you end up with a protein that's completely non-functional. And then you probably have a cell that doesn't survive. And you can also have mutations that are synonymous. They'll not really make any difference in terms of amino acids. But occasionally, you do. Okay, so what are genes that encode proteins that promote cell division when needed? Guys, remember? Starts with a P. Huh?
Never mind, I was thinking of something else.
Okay. It's okay. Anybody?
What was the question?
Yeah.
Others?
Protein, proto-oncogene. Okay? Okay. So. Okay. Yeah, okay. Anyway, and like I said, I don't like, that presupposes that it's the gene that causes cancer. And if we actually had a gene that causes cancer, selection would weed it out, wouldn't it? It would not be beneficial to the whole organism. So it's kind of a misnomer to say it's a proto-oncogene. It's a gene that is involved in cell replication. That's all it is. Okay, so which type of mutation would cause the most trouble? A mutation that inactivates a proto-oncogene or one that activates it? Yeah. Okay. Now, obviously that once a cell is replicating when it shouldn't be, and there's a gene behind it, that's an oncogene. Okay. Okay, tumor suppressor genes, these are the kind of workhorses to stop cells from deciding, oh, the hell with everybody else, I'm just going to go replicate. And these tumor suppressor genes work during the cell cycle. So there's all these checkpoints in the cell cycle where, so this is in G1 where most cells spend their time. And occasionally they get a signal to replicate because there really is a signal. So that happens. There are these proteins that actually check to see whether, oh, is cell division actually necessary? Or is this some, aberrant signal? Are there growth factors present? You need growth factors for a cell to replicate. Is it large enough? Does it have a sufficient number of nutrients? If it doesn't meet those criteria, those tumor suppressor proteins stop the cells from going on and dividing. Now, there's also a G2 checkpoint. So this is after all the DNA has been synthesized, all the other components of the cell have been replicated, and just before mitosis, there's these other proteins that check to make sure that the DNA was correctly replicated. Is the cell large enough? Are all the components there? If it doesn't meet those requirements, it stops. Oftentimes it'll make the cell commit suicide. It's kind of like someone who, someone is making you play Russian roulette, right? You wouldn't do it on your own. I mean, maybe you would, but you're an idiot. But let's say you're not an idiot, but someone is just going to play a game with you, and if you don't play Russian roulette, they're going to shoot you. So you end up maybe killing yourself, maybe not. Okay, so that's kind of what happens. And then there's also, during mitosis, during the metaphase where all the chromosomes line up along the middle of the cell, there's also a checkpoint there to make sure that the different chromosomes that have been replicated, that they pull apart properly instead of maybe all going in one direction or not. Okay? So the chromosomes attach to the microtubules, which then pull the chromosomes apart to either end of the cell. So there's these checkpoints. It's very stringent. Now, unfortunately, when you get older, things don't work quite as well. That's why cancers are more common among older people. It doesn't mean that you might not have childhood cancers. You might not have, adults get healthy adults seem to get cancers, but it's not as common as for older people when the machinery is kind of running down. So, mutations that activate and inactivate tumor suppressor genes are dangerous. Is that true or not? Which one? What? Inactivate? Yeah. In other words, they stop tumor suppressor genes from producing the correct protein to stop all that mess. Now, you probably heard of this. One of the more common cancers for women, of course, is breast cancer. And like there are these tumor suppressor genes in breast tissue, BRCA1 and BRCA2, among a couple of them. And occasionally someone inherits bad copies. And if you remember, Angelina Jolie discovered she had these variants. The penetrance, in other words, the probability of getting breast cancer, is anywhere from about 20 to 75% in someone's lifetime. So it's fairly high. So she decided to do a mastectomy and then breast recreation. Okay, because she didn't want to die of cancer. So a lot of not all breast cancers, but some breast cancers are the result of a broken tumor suppressor system gene. So when a cell commits suicide, by the way, it's called apoptosis. So there's a cell, and it knows it's all hinky, and it says, I'm going to kill myself. Usually by going out in front of a cop, like a tumor suppressor protein that then shoots it. Okay? So it commits suicide on purpose or by accident. And then when that happens, so here's a normal cell. That's what a normal cell looks like here. And here's one that's undergoing apoptosis. Okay. What if your BRCA1 gene is actually defective? Well, then you've got problems, perhaps. So when a tumor suppressor system is not working, it allows mutations to accumulate. Mutations, remember, are random. Mostly, they either have very little effect or they have a bad effect in terms of not making the correct protein, and the cell that has a mutation isn't functioning properly and dies. Occasionally, it'll have a mutation that keeps it replicating, and there's nothing to stop it. Now, obviously, unregulated cell division leads to the production of a tumor with no apparent function. Now, not all tumors are a problem, some are benign. However, there are some that are invasive, and it's because what they've done is they've lost something called contact inhibition. Okay, so we'll say this is tissue, let's say some organ or something, and you end up with a little cancer cell here and here and here's this tumor. And normally what happens is when the cell comes into contact with another cell, they stop. Okay? There's a signal that tells the cell, oh, don't replicate anymore because you're going to impinge on your neighbor. Okay? But when there's a loss of contact inhibition. Um, Okay, that means these cells then still keep replicating and actually form a tumor in this organ. So contact inhibition. All cells generally have signaling and receptor cells that it can tell whether there's adjoining cells, and adjoining cells also put out certain, yeah?
About the benign tumors, can they, with time, do they remain non-invasive, or with time, if we work, can they become invasive?
A lot of times they do, but sometimes they, of course, they could develop a mutation that is in cancers. Obviously, you'd rather have a benign tumor than a malignant one, but sometimes benign tumors are just as deadly. So oftentimes there's pituitary gland tumors in your brain. They're not malignant, but they're benign, but they're very difficult to get rid of. Okay. So if we remember in terms of cancer progression, we have a tumor. Let's say it could be either benign, that means it's not invasive, or it's malignant. That means it invades nearby tissue. Now, as I mentioned, or as the movie mentioned, sometimes you end up with cells that decide, oh, it's time for a vacation. And they hop a ride, let's say by a freight train, and they get carried to some place, and instead of the train bull beating the hell out of them, actually get off the train, and they find themselves in a tropical paradise. And they're so happy, but they'd be even happier if they had some friends, so they start dividing like crazy, and now they've got a whole slew of friends who are identical to themselves, which is great if you love yourself, not so great if you don't. Okay, so when a cell loses anchorage dependence, so normally what happens is cells have proteins on the cell membrane, and these proteins cause the cells to stick together. You can even get like a yeast cell. Normally are independent cells, but you can get a mutation with a yeast, people who created them, where they end up sticking together and forming like a multicellular colony of yeast. So the same thing happens generally. All of our cells, for the most part, have anchorage dependence, except for a lot of our blood cells and our immune system cells. Okay? And, they ended up the blood vessel or the lymphatic system. Okay. That's metastatic. So when cancer or cells lose anchorage dependence, they take off, and then it's metastatic. That means they're moving and creating new cancer tumors elsewhere in the body. Okay, so this is kind of a schematic, normal cell division, potentially unregulated cell division. You might end up with a tumor that has no effect on surrounding tissue, otherwise it's malignant or metastatic. Now, we've talked about carcinogens, and a whole bunch of them. Cadmium is an element that is cancerous. Barbecue lighter, toluene, an industrial solvent, so a lot of hydrocarbons like gasoline and kerosene and things like that are generally carcinogens. Nicotine, ammonia, paint can have some carcinogens in it. Rocket fuel, carbon monoxide, arsenic, methane, acetic acid. Okay. Also, a lot of these are kind of man-made compounds, but it turns out the plant world is very well defended. And they have plants defend themselves with a lot of chemicals, and some of those chemicals are mutagens and carcinogens. Okay? They either cause mutations, which might lead to cancer, or they definitely cause cells to go crazy. So sometimes you can come across carcinogens from the things you eat. So there's a common compound that plants use. It's a cyanide producing compound, and it's of course it's there to stop animals from eating it. So they eat a bit of it and they get sick and they stop. So if you've ever had an almond that was bitter, that almond had a lot of cyanide producing compounds. It turns out that if you take these cyanide compounds and they do a test called LD50, how much of a certain compound will produce 50% death rate. So they do this, they find out what it is, but they also often find out that when animals that aren't necessarily killed directly by it, they develop lots of cancerous tumors. So it's one of those things that is kind of a general thing about a lot of plant compounds that are, sometimes they affect brain, sometimes they affect metabolism, all kinds of different things. So we're surrounded, in effect, by carcinogens. Luckily, our body does have defenses against carcinogens. So not only does our immune system recognize infectious diseases, but antigens in general. And they can recognize foreign antigens that might cause a problem and then bind to them. Okay, but one of the most common carcinogens is the ones that we purposely take, and that's cigarette smoking. Of course, you smoke a lot of pot and it's just fine. Don't worry about it, okay? Really, I swear. Nothing. Okay. So, anyway, of course, cigarettes have nicotine, but they also have a lot of carcinogens when it's burned. And so you end up with lung cancer, esophageal cancer, throat cancer, mouth cancer. Okay. Now they're saying, oh God, they're saying that it could cause cancer of the colon. I mean, why can't it just be lung? I've always been very careful about what I put in my mouth. Really? Okay. So, then also, of course, there's other reasons. You can get a lot of other bad effects like COPD from smoking cigarettes. Okay. Now, so the risk factors, there's a number of risk factors. First of all, exposure to carcinogens that you can not be exposed to. besides tobacco smoke or any other smoke. UV radiation, low levels of ionizing radiation. It turns out that if you fly in an airplane at 35,000 feet, you're actually getting exposed to a lot more gamma rays than you normally would. Because the atmosphere actually protects us. Also, there are certain rocks with radioactive materials in them, and so you can get exposed to ionizing radiation from that as well. Poor diet. Well, certainly you can start eating stuff that might be a carcinogen, or not getting the kinds of things you need that perhaps might protect you against certain things. And then occasionally you get an inheritance of a mutation in the germline cell. So most cancers happen in the somatic cells. Okay, I think let's take a break here. Okay, so I guess risk factors, of course, lack of exercise, diet, and aging. So I guess that means I shouldn't age anymore. It's probably a good idea. Okay. Now, there's some properties that cancer has that are pretty nefarious. All cells need a blood supply to carry nutrients and oxygen and all that, and to release signaling proteins and hormones, whatever. Okay, so cancer does too. Now, it turns out when our body is developing as an embryo, we obviously don't have genes that specify arm, right? And an arm is composed of muscle, bone, neurons, blood vessels, connective tissue, all that. And all those things, all those different kinds of cells happen during differentiation because of the release of things called growth factors. So when an arm is developing, it's actually an interaction between all those different genes being coordinated and then the cellular environment that they're in. Okay. So it turns out that if we injure ourselves, oftentimes you get blood vessel formation at the site of the injury. Some cardiac patients who've had, let's say, a heart attack, new blood vessels start to form to bypass the blockage, all that kind of stuff. Well, cancer releases growth factors that then create a blood supply with capillaries, blood vessels, and all the stuff that goes with it. And that's why if you've ever seen some end-stage cancer patients, they look like they're down to about half the normal weight. It's because the cancer is using all the nutrients. Okay, so angiogenesis is actually called, that's what cancers do. In many cases, they create their own blood supply. There's some other really terrible things about cancer cells. So remember, these cancer cells originally were part of they're our own cells, our own body, with the same sorts of proteins that identify as cells versus non-cells. Okay, so now we have all these immune system functions that suppress this kind of stuff, but if cancer gets started, one of the things that it actually does is it releases hormones and things that immunosuppress the immune system. So the immune system doesn't come along and attack it as being foreign. So it protects itself from being destroyed by our own immune system. And so these are cancer cell. A dendritic cell is a type of immune cell, T cell. There's all these immunosuppressive molecules that are released. And then there's these signal inhibitors for various kinds of things. Okay, so they inhibit the immune system. Okay, so obviously early detection. Now, most skin cancers, by the way, are really hardly anything at all. They don't usually do anything except for melanoma, and melanoma is bad. It definitely is a metastatic, aggressive sort of cancer. So if you go out in the sun, wear lots of sunscreen. A lot of times, so let's say you have something on your skin, go to the doctor, he takes a little bit of it, that's a biopsy. They look at it under a microscope, see is it precancerous, whatever it is. Okay. Unfortunately, they do biopsies of things that are inside your body too. So there's for prostate cancer, there's this chemical that's released called PSA, and it rises when there's any kind of inflammation or it's damage to the prostate. And before it was like, it was the gold standard, but it's really not, because you can just have an enlarged prostate and have higher PSA levels. So to determine whether it's just normal prostate enlargement during aging or prostate cancer, they'll do a biopsy, where they'll take kind of like a gun that takes pieces out of your prostate. And they don't just do one, they do a whole bunch of them, because it could be that the prostate cancer, it's benign here, it's cancerous here, all over the place. And then same thing goes with like liver biopsy or anything like that. Steve Jobs, of course, he died of pancreatic cancer. Now, you've probably heard about pancreatic cancer as being one of the most aggressive cancers out there. The kind that he had was not. Okay, so the most common pancreatic cancers, you're dead in about 6 months. Also, you don't detect them until they've already metastasized to all over the place. But anyway, let's see, he looks like, he thinks it looks like he's serious and all that. But you have to remember that he was once a hippie. And he believed in all this holistic medicine. And so he decided he didn't like the idea of someone going in and removing these cancers and going under chemotherapy, that he do it holistically, in other words, quackery. And unfortunately, it didn't work, and then he finally went in and had surgery and started chemotherapy, but by that time, it had already pretty much ensconced itself. So he managed to live 7 years, but eventually it killed him. I was a hippie once too, by the way. I swear. You want to see? Okay. Now, there's a whole bunch of therapies for cancer. Most of them suck. One of them is radiation, where you take a beam of X-rays, really focused beam, and you shine it on the cancer. It damages the cells. When those cells get damaged, the immune system, the tumor suppressor system, comes on board and destroys those cells. Sometimes it's done after surgery. Sometimes it's done when you can't get to it with surgery. Sometimes it's done in conjunction with like chemotherapeutic agents. So, like I said, it damages the cells and the DNA, and they commit suicide. Now, some cancers are because there's an ineffective tumor suppressor system that's been damaged for whatever reason. And so instead of gobbling up these cells, it doesn't do that. So you can't actually use radiation because you'll be damaging cells, but they won't be gobbled up. Chemotherapy, you know, that's the kind of stuff where your hair falls out, you feel terrible. Sometimes. It's basically standard chemotherapy. You are poisoning the body and hoping that it kills the cancer faster than it kills you. And sometimes it doesn't work that way, unfortunately. Oh, it's also oftentimes you use a combination of them because you can attack cancer along different biochemical pathways, along things that most normal cells wouldn't be expressing. Chemotherapy also tends to suppress your immune system as well. There's also targeted chemotherapy that just attacks cancer specifically. So there's Herceptin, and if you have this oncogene HER2, it's very effective against it. It may not last because, as you remember, cancer cells keep mutating, and you might eventually come up with a mutant, not because cancer wants to, but just by accident, that is actually blocks the function of Herceptin.
So it becomes receptor resistant?
Yes, exactly. Also, now standard chemotherapy kind of works, can work on many different kinds of cancers, and if you start varying the agents, you can handle like a cancer with lots of new mutations. But targeted chemotherapy, unless you knock out all the cancer, it's likely to result in resistance. Okay, now, as I mentioned earlier about cancer causing immunosuppression so the immune system does not attack the cancer. Well, there's a lot of new stuff out to get the immune system to actually attack the cancer. So it's immunotherapy. Obviously, you have to overcome a lot because this cancer is a thing that's actually suppressing the immune system. So one of them is a checkpoint inhibitor, like the Jimmy Carter, former president, had brain cancer. They put him on a checkpoint inhibitor, and he's in remission. He doesn't show any signs of cancer. There's these antibodies that you produce against the specific kinds of cancer. There's also one of the best ones now, this is coming into use, is the immune system recognizes, let's say, foreign infectious particles, let's say bacteria or viruses or whatever, because of certain antigens. And so like when you get a vaccine, basically you're getting these antigens, your immune system ramps up, starts to recognize them, and then remembers them so that the next time you come in contact with that, it's already ready to go and you won't get sick. Well, that's not quite the way it works with cancer. What you do is you take some of those same antigens and then you put them into these dendritic cells as RNA, and you're actually kind of making the cancer think, well, you're trying to make the immune system think that the cancer is actually like an infectious disease. So it'll start immune response to it like an infectious disease, just like, you know, if you had, let's say, streptococcus or something like that. And so there's a lot of, now some of these work, some of these don't. But I think they're getting a better handle on it, so certainly more people are surviving certain kinds of cancer. This guy died. He's an actor. So he went into remission. That's when, you know, you don't have active disease. Why is ovarian cancer so deadly and maybe not prostate as much? Well, it's detection. Prostate cancer, usually a guy will have trouble peeing or having to pee many times in the middle of the night, difficulty peeing, and they'll go into his urologist and they'll check him out and see, oh, he's got prostate cancer. So, and prostate cancer is fairly slow growing. Well, ovarian cancer, usually it's much like pancreatic cancer. You don't see it until it's too late. Instead of metastasizing through the blood vessels, it uses the lymph system, which is part of the immune system. I already told you about cancer, but now there are a lot of viruses. There's many different things there's carcinogens, all that kind of stuff that cause mutations. Well, there are some viruses that actually cause cancer too. Viruses take their genetic material, put it into the cellular machinery of another cell, and then that cell starts producing copies of the virus. Well, human papillomavirus is a sexually transmitted disease, but they actually have a vaccine for it. Now, there was a kind of controversy because it's a very underutilized vaccine. A lot of religious people don't want their daughters or sons to get it because it would be like them saying, oh, it's okay to go have as much sex as you want. It's not really true. So they don't get vaccinated. And then, of course, they live someplace like Midland, Texas, where there's nothing else to do but have sex. And they get genital warts, and eventually they get cervical cancer, and they die. And that's just natural selection. That's selection on being stupid. Okay. So anyway, men should also get it too, because not only can you get it, you know, in the cervix, but if any of you have ever tried oral sex, well, you can get it that way too. Okay. So to not have that happen, you might want to get vaccinated. Okay, so remember, somatic cells are the regular cells of our body that have nothing to do with the germline cells that result in gametes. Okay, so all those kinds of mutations are not heritable. But some mutations that cause cancer are heritable. They're found in the germline cells. A good example is a cancer of the eye called retinoblastoma. There's two forms. There's a sporadic form. Now it's a recessive, so you need two copies of the mutated gene. So sometimes there's sporadic, where it has nothing to do with the inheritance, and it's very, very rare. But all of a sudden, you have one individual who gets a retinoblastoma. It's usually only in one eye. However, there is an inherited form where you get a copy, a mutated copy, and then one of your retina during copying itself gets another similar mutation, and then suddenly you've got retinoblastoma. So if you inherit a copy in all your cells, then it's likely, first of all, that you're going to get it. The penetrance is, that means probability is about 90%. And the only way to treat it is to remove the eye. And someone who actually has a hereditary form, they're almost sure to get it in the other eye as well, so they end up blind. There's a famous guy named Daniel, I think Fish or some, what is it? I forget. Anyway, he's blind. He got retinoblastoma. So, and he happened when he was a little baby. Well, he did something amazing. He started making noises, and he'd actually pick up an echo of those noises off the walls and things like that. And he got really good at it. In fact, he started doing these little clicks that were very effective. You could tell what something looked like, because you get an image like sonar, right? Or like what bats do, echolocation. He can tell texture. He can ride his bike.
Was it Daniel Kish?
What?
Daniel Kish? Daniel Kish was that his name?
Daniel Fish. I think it's Fish or
Kish.
something like that. You'll see.
It rings.
Put in Batman. Okay. So anyway, he can actually ride his bike. He likes to go hiking. He can hike whether it's light or dark out, because he doesn't need to have light, right? It's like he did it well. He was furious that anyone would think that he couldn't do anything. So he kind of got this mechanism that he found useful and made it better and better. And he started teaching other people it. And so he's been now the thing is, most of the big blind loss of vision agencies and all that, they've been treating blind people kind of like, they have a handicap. And so there's been kind of like, they haven't really joined on with what he's been doing. So there's been some controversy, but it's been really well established what he can do. And the people he's trained, it works better with younger people compared to older people, things like that.
Yeah, if I look it up, it is Daniel Kish.
What?
It is Daniel
Kish.
Kish.
Kish, yeah, that's right, it is, right, yeah, yeah. In fact, okay, this is a BRCA system, tumor suppressor system that goes haywire. It's also associated with a number of other kinds of cancers too. Okay.